My name is Þórir Óskarsson. I'm a PhD student at the University of Iceland and part of the iStopMM research group. And I'm here with the iStopMM group, and we are reporting results from our studies at US this year. We are evaluating the utility of flow cytometry-based assays for monitoring multiple myeloma precursor disease. But the inherent limiting factor for flow cytometry analysis is the quality of the bone marrow sample, particularly hemodilution of bone marrow samples. And plasma cells are largely restricted to the bone marrow, and any blood contamination, or what we call hemodilution of the bone marrow sample, will lead to underestimation of plasma cell number and may even lead to false negative results in MRD assessment. So really a clinically relevant matter. And we don't have any gold standard measure for hemodilution. But with flow cytometry analysis, we don't only get information about the plasma cells, normal and abnormal plasma cells, we also get a general overview of the cellular distribution of the bone marrow microenvironment. And what we wanted to do, and is part of a study we are introducing here at US, is see if we can use the distribution of bone marrow associated cell population to really evaluate the degree of hemodilution in a bone marrow sample. And what we did was we analyzed a large number of first pool bone marrow samples and second pool bone marrow samples, which are generally more hemodiluted than first pool samples, and they used that as a reference group for optimal and suboptimal samples to evaluate the predictive power of different bone marrow associated cell population of hemodilution. And based on these findings, we developed a hemodilution scoring model which incorporates our strongest predictors in a single linear model which can be used to evaluate the degree of bone marrow hemodilution in diagnostic bone marrow samples. And this hemodilution scoring model can really aid in determining the quality of bone marrow samples, and they help us to decide whether they are fit for diagnostic use or not. Importantly, this is data from diagnostic samples, and it has not been and the model has not been evaluated in a treatment setting. So that's what we would want to do next in following up this study, is to evaluate if this model is applicable in every setting, in MRD setting, in diagnostic settings. So, I'm really interested if we can use this technology also to evaluate disease status and monitoring of precursor conditions in peripheral blood. So there are a few abstracts here that are focusing on circulating tumor plasma cells, and I think that's a really promising way of circumventing this issue of hemodilution, is moving this assay from bone marrow samples to a blood-based analysis.